Hello, I am Guilhem Sommeria-Klein. I am a postdoctoral researcher here in the Turku Data Science Group. I am a modeler. I am doing statistical model to understand microbial ecology, and I'm applying these models to data collected from different ecosystems. These data are originally DNA that has been amplified and sequenced, and this gives a wealth of information on the diversity of microbial ecosystems. And microbial ecology consists basically in asking who is where and why and doing what. And there are many different ways to look at this question from these data that are coming in larger and larger amounts. The way I have been looking at them with my collaborators is more focusing on large spatial and temporal scales. So one example is oceanic plankton. So oceanic plankton is made of many different types of microbial organisms that have in common not to be able to swim actively, but they are just transported by currents. And so these communities are very diverse. They have a lot of interactions going on, although they are just being passively transported by currents all the time, all across the globe, and they are being submitted to very different environmental conditions. So it's very fascinating to have this functioning community, a highly complex functioning community, although they live in such an unstable environment. And there is indeed a lot of temporal instability locally, but we have shown that actually at the global scale there are seemingly quite stable geographic patterns emerging. And using statistical model, I've been trying to disentangle what is the contribution of different factors to the shaping these communities at the global scale and these geographic patterns in their composition. Another example is communities that are associated with host organisms. So microbial communities are associated with, basically all larger organisms, being from plants to mammals, and also all kinds of larger organisms. Of course, in this group there is a lot of focus on studying microbes and microbial communities that are associated with the human gut or skin, for instance, and how the composition of these microbial communities and their variation in time relates to the health and disease. But in my case, I am looking at this variation over longer time scales, and wondering how variations on longer time scales contribute to shaping these microbial communities. And to do this, we look at different hosts that are more distantly related to each other, that can be placed on evolutionary trees, like here for mammals. And we are modeling how the composition of the microbial communities changes over long time scales. And doing that, we've been able to show that there is actually quite a lot of the variation that can be observed across different host species, for instance in mammals or birds, can be explained by long-term changes. So these changes, they can be linked to the fact that microbes are transmitted from one generation to the next, but also to the fact that the features that there are features that actually constrain the composition in hosts, and those are also changing slowly over evolutionary timescales. And we are building models to try to disentangle different factors over this different timescale that shape the current diversity of microbial communities across different host species, but also in the future we're also interested in looking at this across different human population at the global scale. So there has been recently a lot of work on linking specific bacteria, species or strains to a specific disease, but there is a lack of background on the overall variation in human population in their microbiota composition, and what are the reasons for these variations. And so having a better understanding of the ecological backgrounds of these variations, why they are occurring and at what time scales, will help contrasting the changes that may be linked to recently introduced over evolutionary timescale diseases and from background variations